as my clinical work, I do see a lot of those patients, and we've been interested in developing techniques to assist in the prognostication at an early stage of these patients for many years now. And this is really collaborative work, not only by myself, of course, but with the intensivists in several hospitals in the Netherlands, in particular the Medisch Spectrum Twente, and several collaborators in hospitals and here at the University of Twente as well. Before I go into detail about the role of the EEG, this is my disclosure, I'd like to recap some physiology that hopefully will assist in appreciating why the EEG is such a good method to assist in the prognostication. Shown here on the left is a movie clip of neurons exchanging information. The bright whitish blobs represent the intraneuronal calcium that is very shortly before a neurotransmitter is released, increases tremendously in concentration. So indirectly, we're basically looking at neuronal communication at this moment. And this is what neurons do all the time. And hopefully it makes some sense. That is not an observation you can always do if you talk to people, by the way, but that's would be the topic for another kind of seminar. Now, in order for this process to occur, a lot of requirements have to be fulfilled. And I often use this cartoon showing an action potential. That's the little guy on the top, and the two neurons on the bottom, neuron A and B, that like to exchange information. Now, in the neurons, this is typically typically realized by the propagation of action potentials, as illustrated here. And our little guy quickly moves along the axon with velocities between a few meters to 60 meters per second. And then at the chemical synapse, which is one of the most important structures in between the neurons, it becomes typically a little bit more difficult. But if neurotransmitters are excreted in the way they should be, and there is sufficient input, excitatory input, for instance, at the receiving neuron, then eventually this neuron will also generate an action potential. And this is basically what neurons are designed for. And I often compare a neuron with a polling station. So neurons are continuously bombarded with yes and no votes. And the votes are counted. And if there's a surplus of yeses, then the neuron will typically generate an action potential. And if there are more no's than yeses, then the neuron will not. So, and this integration occurs at the somatic part of the neuron, amongst others. And the nice thing is, if you have a lot of neurons chatting in this way, exchanging information, then it allows us to measure part of that activity, in particular from the neurons in the cortex, with an EEG. So apparently this continuous counting of the votes is associated with the intraneuronal currents and giving rise to periodic rhythms, as is illustrated here on the top. And so essentially with an EEG, we are listening to the communication of the neurons, and in particular the neurons in the cortex of the brain. And that appears to be very relevant also because the cortex is not only the most interesting part of the brain, but it's also the most vulnerable part of the brain as it comes to energy depletion. And that's where we are talking about today. Because patients after cardiac arrest suffered from a temporary energy depletion to the brain. And that is quite directly reflected in the rhythms, as we will shortly see. An important part, of course, is that everything needs energy, and that applies to neurons as well. So our little guy needs oxygen and glucose in order to function properly, to be able to communicate. And that is generated mainly by the mitochondria, using oxygen and glucose to generate ATP. And this ATP is indeed needed to realize neuronal function. And the natural question is, which processes of these neurons do need most of the ATP? And is there a gradient, for instance, in processes that fail if ATP availability is limited. Now, this has been studied by several groups, and mainly by the group of David Atwell, using various calculations and data from experiments. And what he showed is that about 20% of the ATP is needed to maintain resting membrane potentials. About 20% is needed to restore the ion fluxes associated with action potential generation. There's also energy needed for maintenance and repair. But a very large part of the ATP consumption is needed for the chemical synaptic transmission. And the total is about 20 to 25 watts. That's the amount of energy that adult brains need to function properly. And of course, what becomes now interesting for the clinic is what happens with all these processes if ATP depletion, if there's ATP depletion, a limited supply of ATP. Will they all fail at the same moment, or will there be a gradient? And that is what I would like to discuss shortly now. Now, the observation is, so this is essentially already a summary of the answer to the question I just posed. In essence, this is the situation that occurs if there is a limited availability of glucose and oxygen. It is shown from experimental data that this process does not fail at the first moment. So neurons still maintain membrane potentials, they can transport action potentials, but the chemical synaptic transmission is halted very soon, actually. So this becomes very difficult. Allele guy cannot communicate, jump the river, so neuron A cannot communicate with neuron B because the, the chemical synapse is not functioning properly. And is that really true, actually, what I just said? Well, we have, as I just said, several observations from in vivo research and in vitro research, but there are also some observations more at the macroscopic scale that indeed show that dysfunction or temporary dysfunction of synaptic transmission is the first process that is observed if ATP availability is limited. And what is shown here is an observation in a human, in a patient that was referred to our department, showing an EKG in red and above it in black part of an EEG, just one channel. Left to right is 10 seconds. And we see a nice alpha rhythm amongst others in this recording. And it happened that in this particular patient who was referred for either syncopes or seizures, that during the EEG registration, there was definitely an abnormality in the electrical activity of the heart. In fact, there was a complete cardiac arrest. And what is interesting to see is that for the first 5 seconds, there's no change in the EEG. And for another 5 seconds, there's hardly any change. There's one ectopic beat maybe here. And then the EEG slows down after about 15 seconds. And about 20 seconds, the EEG becomes flat. And you may wonder why we didn't start resuscitation immediately. Well, in fact, our lab technicians are trained such that they can wait a little bit to see what happens to the patient before they start resuscitation. In this case, the patient had suffered various of these events, so the likelihood that this would not resolve spontaneously is very small. So the lab technicians remained cool and even wrote down what they observed in this patient: eyes open, turned upwards, no response. So definitely no conscious at that particular moment, but very quickly the heart started again and EEG rhythms come back. Now this is a macroscopic observation that supports the notion of temporary synaptic arrest after cardiac arrest. And it's also shown that already within 30 seconds, the brain function stops in these patients. And it is also shown that if you start the heart soon enough, there's no residual damage done. Everything functions properly again, no neuronal damage. Now, clearly this is not massive neuronal death, otherwise we wouldn't have the observation that the patient is responsive again without any neurological deficits. This illustrates a significant dysfunction of the synaptic transmission. Clearly, if this would last longer, say several minutes, 5 to 10 minutes, then the synaptic transmission is probably not the first process to fail, and it may not be completely reversible. And it has been shown that indeed, after 5 to 10 minutes, irreversible synaptic damage may kick in. And there's also selectivity. Not all synapses have the same sensitivity to ATP depletion. Some can stand it for much longer than 10 minutes, and other synapses cannot. So there's a lot of heterogeneity, selective sensitivity to ATP depletion already at the synaptic level. Now, if the ischemia becomes longer or more severe, then it's not only the synapse that will fail. There also will be a problem with the maintenance of the membrane potentials and therefore also the generation of action potentials. So that is actually the second process to fail after synaptic depression. And there's a lot of literature about it. I just want to show one observation with you. And that's an experiment that had been done a couple of years ago in Nijmegen, where colleagues used rats for their epilepsy research. And they also had eventually to terminate the experiment. And they did it with decapitation, which is relatively a humane way to end an animal experiment if the animal is small enough, clearly. And at the same time, they recorded electrical activity of the brain. And what they observed is shown on this slide. This is a summary of 9 rats. And initially, the rats are awake, and there are some EEG rhythms. And then at T0, there's this artifact, which is the decapitation artifact. You can observe that for about 5 to 10 seconds, there is still in most of the rats electrical activity. And then after about 20 to 30 seconds, there is electrical silence. Similar timescales to human, I think. But then after about 40 to 50 seconds, there is a huge wave, which was called the wave of death by these authors. And thereafter, there's electrical silence again. Now, at the time, there was a lot of speculation about this wave. But the interpretation is relatively straightforward. This is massive neuronal depolarization in the red brain that occurs about 1 minute after the decapitation, reflecting loss of membrane potentials. And if there's a sufficient number of neurons where that happens synchronized, then this wave can be observed, as is shown here. And actually, one of the PhD students was able to simulate this very well in an in silico model. So loss of membrane potentials is a second part in the cascade of ATP shortage, looking at the neurons. Now, finally, and this is already associated with loss of membrane potentials because that is associated with massive ion fluxes, destroying actually the physiological ion gradients. And that is associated with changes in osmolarity, and neurons will then eventually swell because of the influx of water. And by the way, this can still be reversible, as we discussed. Depolarization can be reversible in some circumstances. Even cell swelling can be reversible. Clearly, if it is such a severity that the membrane is actually disrupted by the mechanical forces, at least that is one of the events that can be observed, then it is irreversible. Now, this later process has been studied in detail in a very nice paper recently from colleagues in the US, Brian McVigar, and they were interested in what critical ion fluxes are associated with the occurrence of cell swelling. I will show you 2 movies. On the left, they measured intraneuronal sodium concentrations with a dye after the chemical induction of events very similar to ATP depletion. And what is shown, I will start the movie, is that, let me turn it back a little bit. Initially, all the neurons are blue, reflecting a low intraneuronal sodium concentration. That's physiological. During physiological conditions, the intraneuronal sodium concentration is about 10 millimolar. But after application of a particular drug, in this case a drug that keeps the sodium channel open, but it's very similar to ATP depletion because there's massive sodium influx. And this is indeed shown here in the movie in a very nice way. So all the yellow indicates a huge increase in intraneuronal sodium concentration. And at the same time, it's observed that the neurons increase in size. So there is cytotoxic edema. And at a larger scale, this is observed as well. This is hippocampus. And the same drug is applied, resulting in massive influx of sodium, veratridine, and then you observe this huge increase in volume, reflecting cytotoxic edema. And so this has happened. This happens at the third stage after severe ATP depletion. The time is too short to go into detail about the cytotoxic edema, but in essence, there is a massive sodium influx and chloride influx. Both ions need to go into the cell. Electron neutrality needs to be preserved, and that results in a huge osmotic gradient, resulting in the influx of water and the associated increase in cell volume. So how is this reflected in the clinic? Are there particular measures and readouts that correlate with these events? Well, firstly, if the neuronal dysfunction is limited to a dysfunction of the chemical synaptic transmission and or electrical phenomena only without associated changes in the cell volumes, an MRI scan will not show any abnormality, at least not an anatomical MRI scan, because it mainly measures either cell breadth or changes using a diffusion-weighted MRI in the relative ratio of neuronal volume and extracellular volume. Now, if there is a change in the cell volume, then indeed we can find changes in the, for instance, using a diffusion-weighted MRI. Electrically, however, it is observed or known that the sensitivity is much larger, because already when chemical synaptic transmission fails, this is reflected in the EEG. As illustrated here. And the same applies, of course, if the membrane potential is lost and cell volume regulation is disturbed. But as the EEG mainly reflects synaptic transmission, it is very sensitive to sense changes in neuronal function resulting from ATP depletion. And this is one of the reasons why the EEG is such a useful instrument in patients in the ICU, amongst others, patients who suffer from a status after cardiac arrest. Now, this is how this looks in our ICU, in our previous ICU. The patient submitted to the intensive care unit and typically being treated with mild therapeutic hypothermia, because fever is probably detrimental in these patients, so we try to prevent that. And so most patients are sedated. We prevent fever for about 24 to 48 hours after cardiac arrest, and then we taper the sedation and hope that the patient regains consciousness. But since about 5 to 8 years, we monitor this process with a very early EEG. And this is the infusion pumps. This is always standard in all ICUs around the world. This is the ventilator and monitoring of the cardiac and hemodynamic function. And this is our EEG monitor. So what can we observe in these patients? Well, it's good to realize that in essence, in all these patients, immediately after arrest, the EEG is flat, as was illustrated by the patient who went to our department for either syncopes or seizures, because already after 1 minute of cessation of heart function, we observe a sufficient voltage of synaptic transmission that the EEG doesn't show any electrical activity. So actually, this is the pattern we observe in all patients if we would record very soon after the cardiac arrest. Of course, we do not know at that particular moment whether this reflects only synaptic failure and or neuronal loss of membrane potentials, or also cell swelling. However, we can buy time, and then the EEG will provide us, hopefully, with information about the underlying structures that have become damaged. Indeed, time will tell. And this is an example of a patient already recorded several years ago, showing 3 epochs of an EEG, 3 hours, 10 hours, and 27 hours after a cardiac arrest, showing a relatively favorable evolution of the EEG. So this is 3 hours after arrest, essentially an ICU electric recording, some return of rhythms, and more return of physiological EEG rhythms 27 hours after arrest. This actually is much better even. Another patient, 5 hours after rest. It's mainly EMG activity, I think, that we observe here. And then 12 hours after rest, suppression burst, and 20 hours after rest, a complete continuous EEG. And this is a very favorable evolution of the EEG in patients after cardiac arrest. So if this is observed about 20 hours after rest, as in this example, we can reliably conclude, that has been shown now by many studies, that apparently most of the damage done was irreversible and a reflection initially this EEG of synaptic arrest. So we started with some of these pilot observations about 6-7 years ago, but by now we have monitored in our hospital, I think, more than 700 patients, and we have a huge database, probably the largest of the world now, from collaboration with many other hospitals in the Netherlands, in particular the Rijnstaat ziekenhuis in Arnhem. I think MST and Rijnstaat already have maybe 800 or 900 patients, and in collaboration with many other clinics in the Netherlands, and actually one in Belgium as well, we have over 1,000 observations right now. And this has assisted us in proposing a classification of the EEG in these patients and making very reliable predictions in this particular patient group about the prognostication. So we decided to use essentially 6 categories of the EEG. One is isoelectric and 2 is low voltage. And on the other end of the spectrum, we have normal or continuous EEGs with normal amplitudes, the diffuse slowed EEGs, and the relatively a little bit more complicated category 3 and 4, where the EEG is discontinuous, showing burst suppression patterns. Time is a little bit too short to go into details, so I refer you to the literature, but if we keep this particular classification in mind, we can define about 50% of the patients, identify about 50% of the patients, either with a good or a poor outcome within 24 hours or up to 24 hours after cardiac arrest, which is a tremendous increase as compared to about 7-8 years ago, where the only test that we had for prognostication was the somatosensory evoked potential that assisted us in identification of patients who did poor with a sensitivity of approximately 30%. And on this slide, I summarize the key findings, not only from our work, but also from many others like Andrea Rossetti and Spalletti and Sivarayu, other clinics that essentially keep the same categorization of the EEG. So this would be T0. We have a flat EEG recording. And if after 12 hours after cardiac arrest, we observe a continuous EEG, not low voltage, continuous patterns, and that's irrespective of the frequency, then those patients do neurologically very well. And the sensitivity of that observation ranges, it's probably closer to the 50% than to the 30% is indicated here. So approximately 50% of those patients can be identified in this way. And if this is observed, then the patient has a very high likelihood of a good neurological recovery. And the specificity of this observation is close to 100%, and the sensitivity is about 50%. So we can reliably identify patients with a good neurological outcome already 12 hours after arrest. On the other hand of the spectrum, we have patients that have a persistent flat EEG recording, and if this is observed at 24 hours after arrest, that is a too long time scale to hope for sufficient synaptic recovery. Then there's much more irreversibly damaged than only synapses. So this is indeed associated with a significant neuronal death eventually as well. And the same applies for a particular burst suppression pattern that we labeled burst suppression with identical bursts, where the bursts within a channel are essentially copies of a previous burst, which is a very peculiar burst suppression pattern that thus far, to my knowledge, has only been observed in patients after cardiac arrest. If that pattern is observed actually at any time, but it's typically observed within 24 hours, then the neurological outcome is also poor. And we define poor with the cerebral performance category scale. So CPC 1 and 2 we define as good. The patient can return home, or he can return home with a little assistance, which is CPC 2. 3 is nursery home, 4 is minimal conscious state, and 5 is death. And we and many others consider 3, 4, and 5 as a poor neurological outcome. So this allows us the early EEG, and please note that this is during sedation and during treatment with mild therapeutic hypothermia, that the early EEG can reliably identify approximately 50% of the patients. So 50% of the patients with a good outcome and about 50% of the patients with a poor outcome. At the same time, of course, this still leaves room for improvement because there's still a significant fraction where we cannot prognosticate reliably at such an early stage. So to just show a few examples, this is a patient 4 hours after arrest, sedated with propofol and remifentanil. The main observation in the EEG is an AKG artifact. So it's essentially flat, but this is too early to make a final conclusion. So you have to wait. This patient can still completely recover because 4 hours after arrest is a too short period. This is still within the possibility of complete synaptic recovery. This is an example of a burst suppression, but non-identical bursts, which can be, amongst others, be mainly induced by propofol, for instance. So this is unclear at this moment, how this is the patient in the gray category. So this could be propofol only, or a combination between propofol and brain damage. This is an example of burst suppression with identical bursts. So this is epoch 1, epoch 2. There can sometimes even be a time in between of minutes. But if you observe the bursts in the same channel on subsequent time intervals, it can be noticed that the shapes are very similar within the channel. And this is known as burst suppression with identical bursts. Historically, actually, it was already observed by Hughes in 1986, and he called it stereotypical bursts, and he described 3 patients. So it was sort of a reinvented by us or rediscovery, but we've been able now to see it in many, many more patients than only 3. And also in the Hughes paper, he observed it only in patients after a cardiac arrest. So this appears to be pathognomonic for severe cortical damage. In fact, we have shown, but time is too short during this seminar, that on post-mortem, you can indeed find significant neuronal damage in actually all brain regions in patients showing this particular EEG pattern. This is another example 4 hours after arrest. Now, a very nice EEG. So this patient has shown very quick, good neurological recovery already 4 hours after arrest. And this is a pattern we observe quite often as well, actually. So this is very, very favorable. And finally, an example, 24 hours after rest, still isoelectric recording. So this is prognostically unfavorable. There's one disadvantage, although it's not extremely complicated to look at these patterns, but there is one disadvantage still, that you do need a human to interpret the EEGs. And that's typically a neurologist, neurophysiologists with some expertise or quite some expertise in interpreting these signals, often, of course, assisted by a lab technician. And we and many others have tried to make algorithms to assist in this process. And there's many, many approaches, of course. The first one is, for instance, extraction of explicit features like the mean amplitude, variance, frequency, correlation between channels. So many traditional techniques used in machine learning. And then classifiers ranging from decision trees to random forests, for instance. So that's an approach we indeed have pursued as well and are still pursuing. But we also decided that maybe another approach, in particular deep learning, might work for these patterns. And well, there's a big boost in these techniques, as you know, and we were interested whether we could train a deep net with these patterns and see how well it performed as compared to a human observer, a human interpreter. And also to ease a little bit maybe the burden of the visual analysis that is felt. And of course, we cannot be present 24/7. So what we did, we took epochs from all our patients, and we had about 900. In the latest paper, we used 900, 600 epochs from patients to train a net, and 300 patients to test the net. Actually, it was an external validation. So 600 to train. And what we did is that every hour we took 10-second EEG epochs for 5 minutes. So that's 30 epochs. And we weren't actually interested in what the human being would call the pattern. We associated the pattern, or we labeled it with a neurological outcome, because all our patients have a follow-up of 6 months. So the red in this example, this epoch was associated with a CPC of 5. So this patient did not do well. And all the green epochs were associated with CPC 1 or 2, and all the red epochs with CPC 3, 4, or 5. And we did that, of course, for different moments in time, because it is extremely time dependent. And we tried different architectures, and finally the architecture used was a net from the visual geometry group, a VGGC network, which is a very deep net. And this work was mainly performed by Marleen Tjepkema-Kloostermans and Catarina Lorenzo from Portugal. She's a PhD student, and Marleen is an ex-PhD student. And we fed the net with the raw EEG. So we just filtered between 0.5 and 25 hertz, 90-channel EEG, I think bipolar montage. So it's a 2D convolutional neural net, and that's the input signal we provided to the net, labeled with a CPC score. And as just said, 661 epochs for training, internal, so the testing and the validation, and the remaining 234, there weren't 300, 234 for external validation. And those were actually EEGs from other patients and from other hospitals. So we think this is quite a proper way to evaluate how well such a net can perform. And this is the result. So on the left, the visual assessment by human experts. And this is an example of what we observed at TS 12 hours after rest. So we provided human experts EEGs 12 hours after rest, and we asked them to label it. And from that label, we extracted, based on the algorithm I showed earlier, whether or not the prognosis would be good or poor. And the sensitivity to detect patients with a poor outcome was about 37% from the human observer, and for good outcome, 50%. And the deep net, the same EEG epochs at 12 hours, was able to detect patients with a poor outcome or identify with a sensitivity close to 60%, and a similar performance for the good outcome of approximately 50%. So indeed, a little bit better than the human. I would already be very happy if it was the same. So that was definitely realized, allowing us to essentially use these algorithms to assist in the decision making. And because what is shown here, I think we and others are working on it right now as well, have shown that AI can definitely assist in the interpretation of these EEGs, and can assist the clinician with an advice. Of course, at present, we don't completely rely on AI, but this may change in the future. And details of this work were recently published in Critical Care Medicine. But it does show, I think, the power of the AI. We were quite impressed, actually, by its performance. That without any explicit features, just feeding the net with raw EEG signals, we were able to obtain classification accuracy even slightly better than a human expert. So to sum up, if there's an ATP depletion, there are three important processes that fail in the brain. First, synaptic transmission. That can be isolated and can be completely reversible if the ATP depletion is sufficiently short. Second, we have changes in the voltage, so the membrane voltage and the generation of action potential. And finally, there are problems with the cell volume regulation that result in cytotoxic edema. And when that is present for a sufficiently long time, cells will eventually go into, for instance, apoptosis. Notably, this also holds for synaptic transmission. It is possible that there is irreversible synaptic transmission failure without the other processes. This has been observed. So if the ATP depletion is not zero, but critical for the synaptic transmission, but not critical for ATP pumps to keep functioning, then it is possible that synaptic arrests will be irreversible. That is important to keep in mind. So for instance, normal MRI scans will not exclude a poor recovery. EEG is extremely sensitive for ATP depletion, for oxygen depletion and glucose depletion, as it reflects the most sensitive process that is ATP dependent, which is the synaptic transmission. And also of those cells that are most sensitive, that is the cells in the cortex. Brainstem is much less sensitive for ATP depletion. And it allows not only to track the dynamics, but also to assist in the prognostication of these patients. And finally, I very shortly touched upon it, machine learning, in particular deep learning, can assist, and I'm absolutely confident that it will assist in the near future in the interpretation of these signals in the clinic. So thank you. This is a picture of our research group. Jeanette Hofmeijer has been involved in this work. She's a neurologist in Rijstaat and also a professor here at the university. She does a lot of research also in these patient groups in the ICU. This is Marleen Tjepkema, who did a lot with the deep learning and together with Catarina Lorenzo. And where is she on the picture? Oh yeah, she's right here in front of Marleen, working on deep algorithms for deep learning. So thank you very much for your attendance, and I think the floor is open for discussion. So I give back the word to Rob.
Thank you very much, Professor Van Putten, for your interesting talk. I'm sure that there are some questions from our attendees. We have already received a few through the Q&A button. Please don't hesitate to add more, but I will now go through the questions one by one and try to have them answered by Professor Van Putten. So the first one is by Leo Hogendorn. Great presentation. You explained some of the EEG patterns, but how do you explain the identical bursts?
Yeah, this has been hunting us for many years, Leo, this question. This is not so trivial. The explanation for burst suppression is even not trivial. We have made very simple models. Actually, they exist already in literature, where the degrees of freedom of neuronal populations have become very, very small. For instance, by allowing almost only electrical connections, for instance. And those neuronal populations, you need an excitatory and inhibitory population, and some feedback loops can indeed generate identical bursts. But the full explanation on the microscopic level for these patients, I do not have yet. Globally, you can argue that there is the degrees of freedom have become very, very small. It's like a three-dimensional, very low-dimensional system, the brain. And we have observed in these patients that if you look at the post-mortem, that there is about 50% neuronal damage in the cortex, in the thalamus and other deep nuclei. So there's significant reduction of functioning neurons. But this is still a very global answer I give you. So we don't have a detailed answer to the question yet. What exactly is the neurophysiological substrate of burst suppression with identical bursts? Yeah. So there's room for research there.
Thank you for your answer. Next question is from Hans de Weert. Very interesting presentation. Thank you. Have you looked at potential differences between male and female EEGs?
No, we have not. And yeah, that's actually an interesting question. Yeah, I think that the differences in patterns in relation to prognostication will not be significant, but I'm not completely sure because we haven't studied. So in general, with the observation is that we have about 70%, I think, males, 65 to 70% in the ICU after cardiac arrest, and a small number of females. But I haven't looked in this. Yeah, so we did look at something completely different, whether we could define a male from or differentiate a male from a female, but those were all normal people based on EEGs. So there are differences in EEGs between males and females. Actually, that was a paper we published a couple of years ago using a very large database of 1,500 males and females, where we trained a net with about 1,000 and evaluated with the remaining 500, maybe the total was 1,300, I don't recall exactly. So there are definitely differences between the EEG. Of course, there are many differences otherwise as well, but there's also MRI studies, for instance. But for these populations, I don't know. No, maybe we should discuss this. And yeah, I think actually that the damage to the neurons is so severe that it will be much stronger, those effects, than subtle differences in EEG otherwise. Yeah.
Okay, thank you. Next question is from Victor Royas. Wonderful presentation. Would it be a difference in these patterns in patients with a previous history of seizures?
Yeah, that's a good question. I also almost thought that you would ask with a history of hypoxia, because there is literature also that previous exposure to low energy may protect the brain a little bit. We didn't look into that in detail. I think even if patients would have epilepsy, then the effects of the results and the prognostic value of these EEG patterns would be very small. And yeah, we do observe quite often electrographic seizure-like activity in these patients. And I didn't touch upon that. That is also a practical problem in the ICU. But I don't think, but we didn't look at it into detail. We have, of course, all the clinical information. That there is a significant difference in EEGs between patients with and without seizures. The damage to these patients, to the brain network, is so large that even if there would be some effect, a residual effect of that, I think it would be very small. Yeah.
Okay, thank you. Next question, because in the meantime we have gotten 3 extra questions from Luke Geiben. Can cases where AI and humans have a different opinion be characterized, or are they completely random?
Say this again, Rob, the question. So cases where
cases in which the AI and humans have a different opinion of the EEG, can they be characterized, or are they completely random, these differences?
Yeah, that's a good question. And to be honest, we didn't look at that in detail yet. That is, of course, a natural step where, for instance, AI performs better or it gives a completely different answer. And that's definitely a step that is needed to, amongst others, maybe to identify, particular features, to work on explainable AI, for instance. So I have to admit that I don't have the answer to this question, but it is definitely important in the research. Yes. So thanks for the question. Yeah.
Okay. Yeah. Well, at least the questions can give you some ideas for future research. So that's
absolutely great.
Yeah. Next question from Benjamin Dioma. Would a higher density, for example, 32 or 64 channels, potentially improve the performance of the prediction algorithms? So,
I don't think so. In fact, I think a lower density performs probably as good as. We did a study using 10 electrodes, so about half, not for the deep net, So I'm for the visual assessment, and that was on par with 21 electrodes. So whether deep nets perform really better, I don't know. Actually, it is a question we can relatively easily answer, because of course we can train our net with a smaller number of channels and see how that works. And so thanks for the suggestion, Benjamin. But I think because what we also observe in these patients, that the spatial gradients of the EG recordings is very small. So it's very homogeneous, actually, what we measure. Not in the patients who have a quick recovery. They have continuous rhythms, but they have continuous rhythm overall hemispheres, and there's some anteriorization of the alpha reflecting propofol anesthesia. But even without that information, the fact that the EGs become continuous already after 12 hours would be, I think, sufficient to characterize those patients as having a good neurological outcome. So I think there's limited value of many electrodes in this particular patient population.
Okay, thank you. Another question from Hans de Weert. Would near-infrared spectroscopy add any value for improvement of the specificity?
I don't think so. And we did some pilot testing with NIRS, and as most of you probably know, there's a lot of limitations of that technique, what is really measured. And it could be that it has some value, but I would guess it is redundant at the best. Because the nice thing of the EEG is that you really look at the function. And with NIRS, it is even still indirect. You have some impression of the oxygen, but you still don't, and there's quite some questions about the reliability. But eventually what matters is the function of the neuron. And in that sense, EEG, I think, is closer to that than a NIRS. But once more, we did some pilot measurements in about 10 or 20 patients. We never wrote up the paper, but it was quite disappointing. Yeah.
Okay. Thanks again. The last question for now from David Rand. Thank you for a very interesting talk. Have you looked into EEG patterns correlation with prolonged rehabilitation outcome in patients that did not regain full consciousness?
No, we haven't. And we do study a little bit. Well, what we did study, we mainly work from Jeannette, is the likelihood of developing a post-ICU delirium, or an ICU delirium, but also post-ICU when people go back to the cardiology department. We did recently publish upon that, and in general, it is very strongly related to how poor the EEG patterns are. So the longer it takes for the patient to recover, and the longer it takes for the EEG to become better, the larger the likelihood of a delirium is. For a really long-term outcome in patients that do not recover or recover poorly, I think it should be noted that the number of patients that have a CPC 3, 4, or 4 is very small in our country and in many other countries in Europe. And that has to do with our attitude, maybe you could say, that if we think that prolongation of a particular treatment is futile, we tend to discontinue the treatment. So we do not have many patients in nursery homes, either completely dependent of care or in a low conscious state. This is very different, for instance, from Italy. Spalletti has published about these patients as well, and there it is very different. So we are interested now in the long-term cognitive defects of patients with a CPC 2. And actually, Jeannette has received a very nice grant for that from an advanced investigator grant. So we started the BROCA study, at least we will shortly start that, to look at cognitive effects of patients who are conscious and have a CPC 2. But a lot of them, and even the patient with the CPC 1, by the way, a lot of the patients do have cognitive deficits and problems that are not always recognized and also not always reflected in the CPC 1 or 2 scale, because it is a very, very crude scale. And we know quite some patients with the CPC 1 and 2, Jeannette even more than I, but I met quite a lot of those patients well. It's in many patients, it is not completely without any deficit, even if you have a relatively good outcome. So that is what we do study. And in those patients, we will look at the relation between early EEG in the ICU and even EEG recordings in the ward and afterwards, and MRI findings. But that study will start, I think, in a couple of months. So we will know a little bit more in about 4 years, I think. Yeah.
Okay, thank you. That was the last question, and it's perfectly within time. I would like to apologize for the lack of video from Professor Van Putten. There were some technical issues that we couldn't solve in the meantime, so sorry for that. But I hope you still enjoyed the talk, and I think you did, looking at all the different questions we got. I would also like to point out the following webinars we have in the TMSI Expert Talk series. So next week on June 9th, we have a webinar from Professor Soufiane Boudaoud from the University of Compiègne about the characterization of the neuromuscular system by the analysis of HD EMG signals. So you can still register for that through our website or through the newsletter. So I hope to see you all again in our next webinars. And please keep an eye on the newsletters and on the website for other webinars in this series. Yeah, if you have any questions left for Professor Van Putten or for TMSI, please do not hesitate to ask them via email to info@tmsi.com. And before we close this webinar, I would like to give a last thank to Professor Van Putten for his time and effort for this very interesting webinar. And leaves me nothing but to wish you all a good health and stay safe in these troubling times. Bye-bye.